In the 1950s, it was known that DNA provided the genetic code, but the exact mechanism for protein expression was unknown. Marshall Nirenberg constructed experiments to determine this, along with the help of Heinrich Matthaei and Philipp Letter. Nirenberg and Matthaei were the first to discover that a specific RNA sequence codes for a specific amino acid. In order to do this, the pair added a synthetic polyuracil mRNA chain to a cell-free system that used E. coli cell extract. Then they prepared a test tube mixing one amino acid labeled with a radioactive isotope of carbon with 19 other unlabeled amino acids. This procedure was repeated 20 times, changing the labeled amino acid each time. In other setups of the same experiment, polyadenine and polycytosine mRNA chains were used in the same manner as the polyuracil. After analyzing the results obtained from this series of experiments, Nirenberg and Matthaei were able to identify 3 codons. UUU encodes for phenylalanine, AAA encodes for lysine, and CCC encodes for proline. In 1964, Nirenberg and Letter discovered the triplet binding assay, which proved to be a direct method for determining the sequence of nucleotides in the genetic code. To conduct their experiment, the pair took a small strand of mRNA with a known codon sequence and added it to a mixture of ribosomes and tRNAs that were attached to amino acids. The ribosomes bound the specific amino acid tRNA and the synthetic mRNA. Then the mixture was filtered through a nitrocellulase filter that trapped the tRNA that had been paired with ribosome-bound mRNA in the solution. Unbound tRNA simply passed through the filter. Finally, the filter was assayed to determine the bound amino acid that remained. Ultimately, upon completion of Nirenberg and Letter's experiment, 61 codons were identified and contributed to the creation of the Universal Coding Dictionary used today. I have added a couple helpful links in the description below if you're interested in learning more.